everyone to the second Protein Engineering and Design webinar organized by the journal Protein Engineering, Design and Selection, or PEDS for short. My name is Roberto Chica. I am a professor at the University of Ottawa, as well as the editor-in-chief of the journal. And if I may, I'd like to start by giving you a little bit of background on the journal, as well as this activity that we're participating in today. So the journal has been around since 1986, and since then it's become the default destination for papers that deal with the engineering and design of new proteins for applications in biotechnology and therapy, and also for studies on understanding the fundamental link between protein sequence, structure, dynamics, function, and evolution. And over the decades, this paper has published the work of almost everyone that you may have heard of in this field. And when I took over the journal earlier this year, my goal was to make sure that the journal remained the best vehicle for our research community. And this word community for me is very important because it dictates the initiatives that I've implemented since taking over editorship of the journal, as well as new ones that will be implemented later. So a few of the initiatives that we have recently implemented are the following. Number one, we've moved to an online-only publication model, and this is to prioritize speed of publication so we can get your papers released more quickly after acceptance. And this has also allowed us to eliminate all publication charges. So the only charge that exists at our journal is the optional open access fee. Also, as I mentioned, my goal is to make this journal, or make the journal meet the demands of the needs of the community. And to do so, the first thing that I implemented was to update the Editorial Advisory Board so that we could better reflect the diversity of our community in terms of research interest and other parameters. And moving forward, I want to make sure that we also promote the work of the community in the best possible way. So we are always striving to make your work more visible to everyone. And also, I want to help the next generation of protein engineers. And to do so, we have implemented this, or we are implementing this award for best paper published in the journal, which will be given to the junior scientist responsible for the work, so the student or the postdoctoral fellow. So if you are a student or a postdoctoral fellow and you want to be considered for this award, submit your work with us, and we will be considering all papers that we published for this award. Okay, other than that, I'd like also to mention that we're organizing these webinars throughout the fall, and they may continue beyond if there is interest. But for now, we have a really exciting lineup of speakers that are announced. This is on the website of the journal. And you may have noticed that when I organize these webinars, I try to include a diverse group of speakers. And that involves also giving the opportunity to junior scientists who will be able to present their work that's been published in the journal. So continuing on with that tradition, or that intent of diversifying the pool of speakers, I've decided this time to invite a member of our Editorial Advisory Board to tell us about her work, in addition to the keynote presentation that we have coming up later. So before I introduce our speakers, I'd just like to remind you that PEDS is a journal for scientists and by scientists. So if you have any suggestions or ideas of what you'd like to see in the journal, or how the journal can better meet your needs, please don't hesitate to get in touch with us. I'm happy to hear from anybody on new initiatives. And I would also like to explain to you a little bit how this webinar will work. So we will have two presentations. The first one will be a shorter presentation, about 20 minutes, followed by a question period. And if you want to ask questions, the way to do it is to use the Ask a Question button that's at the bottom of your window. There you'll be able to type your question, and the speaker will be able to see it and respond to you. Feel free to write your question at any moment. It'll be saved in this little window. You can also communicate with others throughout the webinar by typing in that little text box at the bottom right where it says something nice. You can also type things there. However, for questions, please use the Ask a Question button. So we'll have that short question period after the first presentation, and that'll be followed by our keynote presentation, which will last about 40 minutes. And after that, we'll have a second round of questions, and hopefully we'll be done by 12:30. Okay, so that's it for me. Thank you for joining us. I'll start by introducing our first speaker, who is Professor Joanna Slusky from the University of Kansas. As I mentioned, Professor Slusky is a member of the Editorial Advisory Board of the journal, and I'll give you a little bit of background on her training. So she graduated magna cum laude in chemistry from Princeton University, and while she was there, she synthesized superconductors and magnets, and this is obviously very different from which we will be talking about today, but still very cool. Then she moved to the group of Bill DeGrado as a PhD student at the University of Pennsylvania, and while there, she worked on membrane protein design. Then she started her first postdoctoral fellowship, still studying membrane proteins in the group of Gunnar von Eine at Stockholm University with a Human Frontiers Science Program postdoctoral fellowship. And then she came back to the United States to join the group of Roland Dunbrack at Fox Chase Cancer Center, where she also studied membrane proteins. And she's been a professor at the University of Kansas since 2014. And she's established a vibrant research program in protein engineering and design, and she will tell us all about it today. So without further ado, I'll hand over the microphone to Joanna.
Just getting things set up. Okay. You can see my screen?
Yeah.
Okay. Wow. Thank you so much. Thank you, Roberto, for inviting me. Thank you, PEDS, for hosting this. Thank you, all of you, for coming. It was so lovely to see so many friends in the chat. My name is Joanna Slusky, and I'm a professor at the University of Kansas. And today I'm going to talk about enzyme design. Now, usually my lab works on outer membrane beta barrels, and we design outer membrane beta barrels, which is why I've got this great image of us playing cat's cradle with a beta barrel. We work on the evolution of beta barrels to understand how nature designed these barrels, and we try to design ligands for these barrels so we can do things like stop antibiotic resistance. But with an audience like you guys here at PEDS, I wanted to talk about something that I think is one of the most exciting questions right now for protein design, which is how do we de novo design an enzyme? And if you're not already sold on enzyme design, I just want to say enzymes are great. And I use this cartoon not because I am okay with violence. I am against violence. I'm against particularly manager process chemist violence. But I will say that that enzymes are great, that they're essential to industry, and I think that this cartoon illustrates it, that enzymes change molecules. They allow for reactions to happen at regular temperature and pressures that would otherwise require high temperature and pressures. They make reactions go up to 1 million times faster. And they're not just important for industry, they're also super important for human health, right? They do all of catabolism and all of anabolism. Anytime you need to make or change a molecule in nature, you're basically using an enzyme. So if we could de novo design enzymes, that would unlock industry and human health, all kinds of things. So I wanted to do it. I wanted to design enzymes, and I said, you know, it's a really, my position doing work on outer membrane beta barrels where everything's a barrel, this is a logical place to be thinking about enzyme design because 68% of all proteins with barrel structures are enzymes. And you may be familiar with this prototypical enzyme fold. This is a TIM barrel, okay? And you can see its barrel structure there in yellow. But fundamentally, barrels make great enzymes because they make a pocket where you can sequester solvent from substrate or substrate from solvent. And so really, to me, barrels and enzymes, they're like peanut butter and jelly. They go together. So I said, great, let's do it. Let's make an enzyme. But the problem is that it's really difficult to design enzymes, right? So if you think about de novo enzyme design and the amazing successes that you've seen, you might look at protein-protein interactions or de novo structures, and they have something like a 50% success rate. Now, I'm generalizing. There are more and less difficult problems, but they have high success rates. And you look at some of the most amazing de novo enzyme design success rates, and they're more like 2 or 3%. And while those are like wonderful illustrations of where the field could go, at the current state of the art, they're not scalable. Okay, so the thing is, we can all make lots of computational predictions of what will be enzymes, but we can't we can't synthesize and we can't test 100 or 200 enzymes. We need an algorithm that can determine which designs will work and which designs won't work. Okay, so if we fundamentally can't distinguish between an enzyme active site and a non-active site, I think somebody's not muted. If we fundamentally can't distinguish between an enzyme site and a non-active site, then this isn't going to work. So what do we know so far? And there was some great work in the early 2000s about what is the difference, what would allow us to distinguish. So what is it? Well, number one, homology. And this is useful for if you have a homolog, then you know that your active site is basically in the same place as the other active site. Great. But the problem is that for things that don't have homologs yet, which is admittedly getting fewer and fewer, even structural biologists working on the enzyme will not know. They can solve a crystal structure, and then they can argue about which site is enzymatic or not until they do a lot of mutagenesis and complicated biochemistry. So for an enzyme designer, we can't use homology because our proteins haven't necessarily evolved. We could use theoretical titration curves. This is some amazing work from Mary Jo Andrechen that I'm going to be talking more about later, but those can be very helpful. We also have pocket volumes, that enzymes tend to have larger pockets than non-enzymes. There's pre-organization. If you have a crystal structure, then the enzyme site is more likely to have a lower B factor. And finally, distance to protein centroid, that the enzyme active site is likely going to be more towards the center of the protein. So this is all helpful, but still, we don't know what's going to be an enzyme and what's not. And humans in general, they're not great at seeing things, right? And so we said, what if we use machine learning? Machine learning can distinguish between things when humans can't. So if we were going to set up a machine learning algorithm to distinguish between enzymatic and non-enzymatic, then you really have to pick the right question. And I think that the question of what is an enzyme site compared to rest of protein, that's really an apples to oranges question, right? Because you've got, with the rest of the protein, you've got the hydrophilic surface that's not really so much like an enzyme active site, and you've got the hydrophobic core, and that's not so much like an active site. So what are you really learning when you compare those things? But what would be really similar, and what I think would make a great comparison to give you that je ne sais quoi of whatever it is that makes an enzyme, is metalloenzyme site to metallo-binding site, okay? Because in both of these situations, you've got a pocket, you've got coordinating residues, you've got binding geometry imposed by the metal. So let's do this. Let's compare apples to apples, okay? Now, if we were to have some machine learning algorithm that did this, then the hope is that it could do two things. It would be able to say that if I gave you a protein with two metal binding sites, you could tell me which is the enzymatically active one. This should be useful to biochemists. And for protein designers with closer to my heart, if I gave you 10,000 designed putative metalloenzyme sites on the same backbone, could you tell me which will be active? Right? That's what we want. So to make this algorithm, we're going to need two things. We're going to need a really good data set, and we're going to need a really good feature set. So let me just reassure you that we've got a lot of data. So it turns out that if you look at the PDB, and you look at what percentage of the PDB is enzymes, it's about 48%, which is tremendous. And this goes to show, number one, there are a lot of enzymes, but number two, there's a disproportionate scientific interest in enzymes, that scientists are not crystallizing proteins that they're not interested in. So we've got 48% enzymes. It turns out there are 35% metalloproteins. And if we put these two things together, we find that there's about 19% metalloenzymes. And what our goal is, can we compare and distinguish between metalloenzyme sites and metalloprotein sites. Okay, so let's look at what we really did for the data set. We start with the PDB. We've got about 40,000 PDB structures that have metals in them. And we filter to make sure that those are of good quality. And then we filter specifically based on the site quality, because we're not actually looking at the whole protein, we're looking at the site, okay? And then we split things up into a data set that we're gonna train on and a test set that we're gonna validate on. This is extremely important. Even Bart Simpson knows that this is the way to do this, right? Or he did after this exercise. Because fundamentally, if you're going to be able to predict something in the future and not just give you back the information that you already trained on, you're going to need to have something that you never touched when you were training. Okay, so our test set is proteins that were only crystallized in 2018 or 2019. Great, so we separate them out and then we do the most important step, which is determining which of these sites is enzymatic and which is non-enzymatic. And so this is the subroutine that we do it, where we use a hand-curated data set, the MCSA, and we actually hand-curated the hand-curated data set, and then we say, what are homologs to MCSA? Do we have the right location of the active site? Great, it's an active site. If these are not homologs, if there's no other evidence of enzymeness, it doesn't have an ACE suffix or anything like that, it's not an active site. And so what I'm showing you here then are the numbers. We've got green numbers are number of enzymatic sites, blue numbers are number of non-enzymatic sites, and we continue to filter. We look at homology so that we have a really non-redundant set, and then we even look at structural homology locally to make sure that the sites are really different, and we end up with about 900 active sites in our data set and about 3,239 non-active sites and 185 active sites in the training set and, in the test set, and 564 non-active sites in the test set. And just keep in mind, this is asymmetric. We've got a lot more non-active sites than active sites. This is what's called an imbalanced data set, and I'll get more to that later. Okay, so what are our features? So we've got geometry features, right? These are how the metal coordinates the protein. We've got pocket lining features, the number of proteins in the pocket lining and the types of proteins in the pocket lining. We've got pocket features to describe the geometry of the pocket void. We've got electrostatic features to describe the electrostatics of the pocket. And then we've got these Rosetta terms, okay? And it looks like Rosetta is totally dominating, but actually we're only using a small percentage of these terms at a time. So what do I mean by that? Well, Rosetta gives energy terms based on residues, based on amino acids. And we're looking at whole feature terms. And so we basically take these Rosetta terms and we're aggregating them either as sums or as means, and we take them as spheres or as shells. And so at any given feature set, we're only using Rosetta in the sum sphere or Rosetta mean shell or something like that, right? And then of these feature sets, we do 60 different permutations of the sets so that we can do Rosetta sum sphere, but without pocket or things like that to just overall get the best feature set. What you'll notice that we don't use, however, is anything that wouldn't really be helpful for de novo design. So homology we don't use, and secondary structure we don't use, and we certainly don't use crystal structure because we certainly don't intend on getting any crystal structures for proteins on any large scale. Okay, so with respect to these features, oftentimes people say, you know, I would just expect that metal coordination geometry would be all that you need. And in fact, probably by sight, you could tell based on metal coordination geometry. And so the first thing I want to tell you is it's not really true. So if you look at all different metals, right, copper, zinc, magnesium, iron, and you look at enzymatic number of coordinating atoms in green and non-enzymatic in blue, what you see is that some metals have a tendency for more for enzymatic and some for fewer for enzymatic, but also that we don't actually use the identity of the metal as a feature because that's not actually resolved by the crystal structure, and it can be quite contentious. So then you say, well, maybe you could just look at the actual metal coordination. Here's like, for example, trigonal bipyramidal. What if you've got sort of filled and vacant? And what I'm showing you here is for all filled coordinations or all vacant coordinations. And what you see is that, yes, in fact, non-enzymatic proteins tend to have a higher proportion of filled than not filled. But I'll also say that mostly we just have irregular, that we're using what I think of as really the best ways to do this with Find Geo or Check My Metal, and we're not getting perfect information yet. And that's something we're continuing to work on. Okay. But what are features that are really different that you might expect to see can give you good distinction? And to do this, we look at the overlap between the kernel density estimates. And so these are the lowest overlap of kernel density estimates. And what we have is 3 of these are Rosetta terms. We've got 2 salvation terms from Rosetta, 1 van der Waals term. LK stands for Lazaridis-Karplus, so this is the EF1 potential, and LJ stands for Leonard-Jones. This is van der Waals, or the attractive part of van der Waals. But what you'll notice for these Rosetta terms is that they're all for 9 angstrom sphere sum, which is to say that they're all basically taking advantage of the fact that enzymes tend to be in the center of the protein and have more amino acids around them, and giving a score based on that. What we also have is pocket volume over here, and that's something that the Thornton group already identified. There's a different distribution where you do tend to have somewhat larger pockets for enzymes, although it's still not completely predictive. As you see, it's only 39%. It's still a 39% overlap. So, okay, what does our training do? Okay, so we came at this really agnostic about what algorithm to use. We just took scikit-learn and we said, great, here are 14 algorithms. Let's do those. And so why am I showing you so many different dots? Well, we've got the 14 algorithms. We've got 14 algorithms, each doing 60 feature sets. And then we're actually optimizing all of them 4 times, once for accuracy, once for precision, once for MCC, which I'll tell you what it is in a second, and one a multi-term where it uses accuracy and MCC and a Jacquard index. So what is this MCC? Because usually you would expect to see accuracy. So why don't we use accuracy? Well, accuracy is actually really misleading for imbalanced data sets like what we have. We have almost 80% non-enzymatic. So if I were to make an algorithm that just said no, it's not enzymatic, it's not, then I would still be 80% accurate. That's misleading. So instead, we're using the MCC, which is basically involves true positive and true negative and false positive and false negative to give an overall truthiness score. Okay, so that's what you have on the y-axis. That stands for Matthews correlation coefficient. On the x-axis, you have precision. Okay, precision is true positives over true positives plus false positives. So it tells you when you're getting it right, are you over predicting correct ones? Okay, so what we're interested in then is not over here where we have all the SVMs, or in the middle over here where we have the neural nets. We want the best of everything. So that's this top corner over here. And if we blow that up, what we see is that the best are these tree algorithms. And specifically, there's an extra tree algorithm that is the best of the best. And let me tell you a little bit about this algorithm, because this is what we're going to use going forward. We're using all feature sets for this algorithm. So we get to see all kinds of information. And this is using mean sphere Rosetta. So when I was showing you before that there was good distinction with things on some sphere, that wasn't ended up being the best algorithm. And this one dot, this one model was converged upon by optimizing by accuracy and by the Matthews correlation coefficient and by the multi-criteria. Okay, so how well does it do? So this model we named Mahomes, which stands for Metal Activity Heuristic of metalloprotein and enzyme sites. Okay, it's also obviously an homage to Kansas City great Patrick Mahomes. And just like Mahomes, we have uniformly awesome numbers. So we have great accuracy and great recall, which is the total positive rate, and we've got great MCC, and we have great precision. And we don't have a lot of things in the literature to compare this to, because nobody else is doing exactly this. But what other groups have been doing is looking at identifying catalytic residues within a protein. Can they identify them? And what we find is that we do quite a bit better, or certainly more uniformly better than these other algorithms, right? The other algorithms will do really well on one metric, but then not so well on another. Okay, so it seems to work. That's awesome. What's driving the algorithm's ability to distinguish? Because usually when we have machine learning and you say, okay, but how does it work? The answer is, look, better is better. It's sort of an engineering approach and we can't say anything. But one of the amazing things about using an extra trees algorithm is that it gives you a metric of feature importance. So let's look at that. So on the y-axis here, we have similarity. That's what I was showing you before. How much overlap is there between the distribution of enzymes and the distribution of non-enzymes? And on the x-axis, I have that feature importance. So we're going to be able to see what things that are more or less similar are actually driving this metric. So the first thing to point out is that geometry, which, like I said, intuitively, I think a lot of us want to believe is going to help, is not helping. No matter how similar or dissimilar, all these yellow dots are all the way on the left, 0 feature importance. Then there were those 3 Rosetta terms that I told you were there for some sphere, and we're using mean sphere, so we don't have those, but we do have 2 Rosetta terms that involve solvation, and those seem to be important and taking into account some of the information that those Rosetta sum sphere solvation terms were giving us. Okay, then I want to point out volume, which here is the least similar feature, but also one that does not seem to be particularly driving from a feature importance standpoint. And I would say that it may be because, although this volume feature is not driving it, we've got these 3 lining features which seem to be taking into account a lot of the information that volume would take into account. So enzymes have here, these are the lining features that we're looking at. We see that there is more volume as defined by the backbone. Enzymes have more side chains nearby. You could see that that would also be part of the volume. If it were bigger, you would have more around it because it would simply be a bigger cavity. More backbone atoms nearby, same thing. More volumes nearby, more volume to the nearby side chains. Again, if you're gonna have more side chains, you may have more volume, and if it's more buried into the surface, then the larger amino acids that are hydrophobic would push that forward. So overall, that's the lining, which I think is making up for the volume to some extent. But what is this? This is some electrostatics feature, and it is the most important feature to driving the ability to distinguish between enzyme and not. So this feature is the second moment of the titration curve of charged amino acids. And this comes out of some work from Mary Jo Andrechin, and her work has continued to use this to better understand the difference between enzymes and not, which is that if you look at a theoretical titration curve for ionizable atoms, ionizable amino acids, I should say, you generally get a sigma, right, with a pKa. That's how you know what it is, like that. But, and this is all the histidines in the TIM barrel, the histidine that's at the active site has this deviation, okay? And it turns out that ionizable residues at active sites have not particularly this deviation, but all kinds of weird deviations in these theoretical titration curves. And the Andrechin group has found that the 3rd and 4th moment of these curves, it really accurately predicts. What we're seeing is the 2nd moment, but I think either way, and what that difference is something that we've been looking into. Either way, this is an important distinction and something that is driving that piece of information. So in conclusion, we have this Mahomes model, this algorithm that can discriminate between metalloenzyme active sites and metal binding sites with high precision and high accuracy and high MCC and high recall. It's doing really well. What's driving it? The 2nd moment of titration curve seems to be critical for these predictions, so it's an important electrostatic concern. The coordination geometry is less important, and fundamentally, I think that this model can be used for enzyme site prediction and protein design. My lab hopes to use it. I hope that you guys will consider using it, and that I hope that we can come to better enzyme design and solutions for industry and solutions for human health because of it. And with that, I'd like to thank my lab, who are amazing, and specifically the really pioneering graduate students who did this work, which are Megan Franklin, who graduated from my lab, and Ryan Feehan, who is still in the lab and continues to generate all of these figures for this talk today. We don't look like this now, we look like this. But I'd also like to thank my funders, Gordon and Betty Moore Foundation, the NIH New Innovator Award, and thank you for listening. I'd love to take questions.
All right, great. Thank you, Joanna, for a very interesting talk. Are there any questions for our speaker? Please remember to type it in the Ask a Question box. Okay, it's going to take a while, so maybe I can start, Joanna, and ask you a question. Okay, so how do you propose to use this information to design an enzyme from scratch, for example, could you then, since you mentioned that the electrostatics are really important, would then the goal be to focus on designing the electrostatic environment in an active site of a metalloenzyme if you're trying to create a new enzyme?
So my vision of this and what my lab is working on doing is not that, although I think that there might be some merit to doing that. I think that, at least right now, there's a lot of technology to make de novo, to make putative enzymes. So let's make 1,000 putative enzymes, and we can rank them by energy and blah, blah, blah. But then ultimately, the best ones of those, let's put it through this Mahomes algorithm, and let's see, is it predictive of enzymes and not? And so, if we can all make 1,000, computational enzymes, then we can screen them and see which ones are more likely to work. That's the vision.
Yeah, and I guess that screening would be pretty fast compared to the design step.
Exactly, much faster.
Okay.
So I'm seeing a question in the box from Giovanna. She writes, "Fantastic work, Joanna. Was wondering if you see a difference between redox enzymes and hydrolytic enzymes." So we don't particularly. I'm going to pull up. I shouldn't have closed my screen because it takes a while to get back into it. But let me get back to my PowerPoint slides because there's a slide for that, which is to say, okay, here we go. So if we look at the enzyme types that are in our set, we find that we have pretty good distribution of the different enzyme types. And Ryan has actually looked at this, and at one point we thought we were seeing different success rates for different types of enzymes. And Ryan, maybe you can comment in the chat box. I think at this point we're not seeing very big differences between enzyme types. Ryan, please comment and say if that's right or if that's wrong or whatever. But yeah. Do you have any more questions? Oh, Ryan says it's right. Awesome. And that's, I think, one of the great parts of this is a lot of the other groups that have tried to predict catalytic ability or not are doing it for a very specific enzyme type. And what we're finding is that there's general features to be had that might really allow everybody to get in on this.
There is a question for you in the Ask a Question box.
Ooh. Okay. Okay, the question is, what are the major issues of false positives, not enzymes detected as enzymes? Not enzymes detected as enzymes. So there were two. So I'll tell you about two of them. Okay. Number one is that with not enzymes detected as enzymes, we've gotten two exciting types of false positives. One is that there are things that, because we're doing the test set in the 2018, 2019, they haven't been hand created by MCSA. And some of the things that are false positives, and we consider them false positives because that's how the algorithm works, were actually positives, right? They were enzymes that simply hadn't been typed yet. That's cool. The second thing is that for a while, we were having a terrible time with the NTPases, that some of the NTPases were getting through as positives and some of them were getting through as negatives. And so those things were showing up as both false positives and false negatives. And we ended up basically having to throw out NTPases because we just realized that the particular flow that we had was not allowing good NTPase recognition. Oh, we've got another one. How large should the training data set, how large should be the training data set to make sure our model is correctly trained? Huh. I mean, you mean this, I guess you mean this particular model of Mahomes. So the way we're training it, right, we're using the number, oftentimes you want about, there's sort of this semi-rule, right? You want about 10 times as many things, as many instances as you have features. So that's something of a rule that you might consider, although I'm sure there are great algorithms that break those rules all the time. But that's something of a rule that people might use. Yeah.
Okay, great. So we're going to stop here. If you have more questions for Joanna, please feel free to keep typing them in the questions in the Ask a Question box, and she'll try to answer by typing it there. All right, thanks a lot, Joanna. Okay, so now I'm going to introduce our next speaker, our keynote speaker, Bill DeGrado from the University of California, San Francisco. I almost feel like I don't have to introduce him because many of you know of his work, his pioneering work. I'll do so nonetheless. Dr. DeGrado obtained his bachelor's degree from Kalamazoo College and then a PhD from the University of Chicago. And there he worked with MLT Kaiser, and he developed the oxim resin for solid phase synthesis, which I didn't know and I find is actually very interesting and cool. From there, he then held a position at DuPont, where he worked, established his research program, and did a lot of really pioneering work on de novo protein design. And this work has obviously had a huge impact on our whole community. A lot of the knowledge accumulated from that period is still being used, by many of us, decades later. And after working at DuPont for about 15 years, he moved to the University of Pennsylvania in 1996. And then in 2011, joined the faculty at UCSF, where he's continued to work in this field of de novo protein design. And that's what he will be telling us today. It's a great pleasure to have you here, Bill. I look forward to your talk.
All right. Well, thank you very much for your kind introduction. Let me start on a couple suggestions. Anyway, let's see. How do I share screen? There it is. All right. So our topic today will be de novo protein design. And we first want to ask ourselves, why would we ever want to design proteins? When we began, we were told we were crazy and it was impossible. And yet we did it because we were seeking a fundamental understanding. Clearly, if we understand proteins, we should be able to design them entirely from scratch. And if our understanding is truly atomic and molecular in nature, we can then springboard from that to the design of smart materials, polymers, enzymes, as Joanna told us, and therapeutics. I'd like to focus today on de novo design of proteins that bind to small molecules and cofactors. This is sort of the inverse of rational drug design. In drug design, you have a protein with a known pocket or cavity, and you try to make a drug that fits in there. Here we are starting with a small molecule and trying to design a protein around it that will bind it. This is one of the great difficult problems in protein design. We've been able to design all types of folds and even proteins that bind to protein interfaces, but it's been much, much, much more difficult to design proteins with deep invaginations that can bind substrates. And when it's done, it generally requires starting with natural proteins, not always, but generally, and really relies on combinatorial screening in the laboratory rather than making the first thing, going to the lab and finding that it works. So can we devise highly accurate and precise methods that do not require building large libraries and random screening? Now, all of protein design really is inspired by the Anfinsen hypothesis. And what he showed was that the sequence in the right milieu will fold to a thermodynamically stable structure, but not only that structure, but function. So sequence codes for both structure and function. Now in de novo design, what we do is to try to design a protein entirely from scratch, rather than by modifying the sequence of a natural protein. And we try to do this in what I'd call a reverse Anfinsen manner. So we're going to start not with a sequence, but instead with a structure. We use computational methods to go from structure to sequence. And Steve Mayo in the last talk in this series spoke a little bit about that process with repacking algorithms. And then we get into the lab and we make it and see if it works. Often it requires a great deal of screening, but not always. Then function is often added more as an afterthought. For example, the first protein that was designed entirely from scratch, both in terms of its three-dimensional structure as well as its sequence, and shown to be folded and adopt the right structure, was alpha 3D. We did that a number of years ago. And then since then, a number of groups have, such as Vince Pecoraro, have built function into it by building metallo sites that are catalytically active. Now, what we'd like to do more recently is to really do the full reverse of the Anfinsen hypothesis. So you'll notice that we consider structure and function together to define the sequence. So we start with a function and let the demands of function really define the details of our structure. That then can be used in more conventional structure to sequence design algorithms. And as you'll see, we can directly obtain function without rounds of experimental screening on yeast and display and directed evolution. So much of our early work focused on trying to mimic various aspects of this amazing helical bundle protein photosystem 2, shown here, which evolves oxygen. And we've tried to design proteins that bind porphyrins, radicals, metal clusters, and over the years had a fair amount of success. For example, we were able to design a 4-helix bundle that binds 2 irons and catalyzes the peroxidase reaction and oxidase reaction, taking this aminophenol to the corresponding quinone imine. By changing the environment of that binding site, we were also able to change its activity so it instead hydroxylates here on the amino group and then subsequently oxidizes further. We've come closer to the oxygen evolving complex, designing tetranuclear systems shown here, in which we have bound waters, second shell, and third shell ligands, all precisely pre-organized in precisely the way we had designed. So we're getting closer. But where we really had been stuck for a very long time, paradoxically, was on the first attempts to design cofactor proteins. Over 2 decades ago, we designed the first heme binding proteins, one being a derivative of alpha 4 and the other being based on a sequence design that Les Dutton and I did over a bottle of wine on the back of an envelope, and turned out to bind multi-hemes within a bundle. And over the years, the group has made tremendous progress in evolving oxygen binding and tuning midpoint potentials. But the structures were very poorly defined from our lab and other labs, and there was really no exact method to not only get structures, but predictably get structures, and that's really what we needed to progress. So clearly there was a piece missing. What was the missing piece? So for years I worked with Mike Therian, and he invited me to Duke. And Dave Baraton's a second collaborator, and this is their graduate student, their graduate student, Nick Polizzi. And we sat down and thought, you know, what are we missing? I'm missing something. And the more we thought about it, the more we thought that the way we design, collectively we, proteins, is that we basically think about what we want to happen. We maybe have a scaffold of a protein and we want it to bind something like a heme here. And so we try to optimize for binding. But if you think about what happens, say, in an antibody, you have, sure, a binding site, but mutations very, very far away are important for the affinity that we see up here. And that is because the protein really completes its folding process when it engages with its ligand. And the completion of the folding really is part of the specificity and affinity that we observe experimentally. So we call that the core principle. And we believe that it's important to design the folded core completely on concert with the binding site if we want to achieve tight and efficient coupling. So we went back and designed a protein that would bind this highly electron deficient zinc porphyrin that's being used for optical reasons. And we start by optimizing its geometry. We can think about building a helix with a histidine that will bind to it, and that helps define the geometry of these two relative to one another. And now we want to build a bundle that will encapsulate it, and we can do this by what we call parametric protein design, in which we systematically vary the geometry of the bundles. Here's the rotation of the helix, and you can see different radii being generated. We can also change the major phase, which defines whether it's a square bundle or more rectangular in shape, and the helical offset. So all of these are in play and help us choose the geometry, in this case searching for one where we get a second we can encapsulate and not clash with the cofactor. Now, a lot of that was done before the most recent work. And what's different now, as I said, is we simply treat the problem of core repacking and construction of the cofactor binding site in one unified calculation, which is done now in a Rosetta with flexible backbone design. So the design of the binding site and the folded core are done in unison to assure that they cooperate with one another. We also avoid placing too many large apolar residues around the bindings, porphyrin, the bound porphyrin, to avoid collapse in the apo state. When we do that, we get incredibly nice NMR spectra for the apo and the holo protein. And Yibing solved the structure rather quickly. And we were delighted to see that the folded core of both apo and holo were essentially identical and very well defined. And yet there were multiple conformations for the binding site, which then coalesce when you bind to the porphyrin. If we cluster, we find two different conformations in the apo, one closed and one opened, and it chomps down like a gator when it encounters the porphyrin. And for that reason, we affectionately talk of these proteins as gators. Again, the structure was defined to the precision of our ability to solve an NMR structure. So on the order of a single bond. Now, what I think this shows is that there's, these are like dominoes where you have each position affecting the other. And if we pull out one of the dominoes, now we really destroy the coupling. So I wanted you to hold on to that picture because it's going to be important when we start to think about designing allosteric sites soon in our talk. So more recently, moving from this photooxidant, we've moved to oxygen-dependent chemistry using a manganese system here, or a natural heme, which is useful if you don't want to have to synthesize the porphyrins. In each case, what the great challenge is that the metal ion is going to change in its charge from by 4 units as it binds oxygen and then progressively moves to water plus an oxo species. So how do we stabilize this dioxygen unit as it goes through the catalytic progression? And Sam Mann decided to put a glutamine here, and that would allow it to stabilize the dioxygen in a linear manner. And it could also flip its rotamer depending on whether it needs to be a hydrogen bond donor or acceptor. After doing design of really a radical redesign, he was able to solve this structure here. And indeed, the second shell, glutamine is there, water's precisely as designed. And so we call it the longigator. And here we can see that it is indeed able to access this manganese 5, highly high valence state, and then transfer that oxo to something like thioanisole. Now, in parallel, Kedeke has been working with heme, which will allow us to do things such as directed evolution if we want to use natural hemes directly from the bugs. And it has succeeded in making a protein that has can hydroxylate aniline. Remember, before we were seeing N-hydroxylation. This CH hydroxylates. Very different reactivity than we saw with diiron. Finally, we asked ourselves, can we put these bundles together? We saw a diiron bundle and the gator bundle. Can we put them together to make a communicating system? And for me, it's easy. I say, hey, here's some cylinders. Why don't we just extend those helices? But Nate Schmidt, who did the work, said it's really not that easy, Bill, because the bundles are fundamentally quite different. The interhelix distance between the two is really 1.6 angstroms, and that's large. And so what he did was to try many different ways to dock the two helices or the two bundles together, moving up and down, and then go to the PDB to look for loops or contiguous chains that would connect the two. And the best solution actually had distorted helices. There's a bit of a pi bulge here in the design. Here's the crystallographic structure of what came out. And here's the overlay. So these are essentially identical at the resolution of the structure, which is on the order of about 3 angstroms. Now, the most interesting thing that we found so far is comparing the enzymatic activity of the di-iron site here. Remember, it has an oxidase activity with and without the communicating porphyrin. So if we have no porphyrin bound, then we get this curve here and follows Michaelis-Menten kinetics. As soon as we add the porphyrin, however, we see that it binds more tightly to the substrate. So Km is improved, but Kcat is greatly decreased. So what's going on? We don't have a crystal structure of the apoform, but we have conducted molecular dynamics simulations in collaboration with Michael Graves' lab. And so he removes the porphyrin, and we see then what happens. And what's particularly interesting is that there's a second shell hydrogen bond that we built in from the tyrosine to the carboxylate. And becomes free when we release this porphyrin, and it can move back and forth between us, like here and to here, preferring the away orientations. And so in this way, we have greater dynamics accessible to the protein and higher turnover. And on the other hand, more clamped down has higher affinity for substrate. So I'd like to pause now and thank my coworkers on metalloproteins. And this is a long-term collaboration with University of Naples, Vincenzo Pavone, Angela Lombardi being the leaders, and many other people, Fabio and Marco, contributed quite a bit to the work I just described. And then additionally, thank my coworkers at UCSF in our group, Nate and Sam and Kedeke, of course, Nick, and Jamie Linkoff in Michael Graves' lab for the MD. And then electrochemistry at Duke and San Francisco University. And finally, the Grand Master phase is Li Jun Liu. So now, can we generalize this to any polar drug? I think you notice that the features we've looked at now involve metal ions, has a high affinity for the ligand, and also flat cofactors. But could we design a protein to bind any polar small molecule, especially ones that are highly functionalized? If we could, we'd have potential applications as toxins, antidotes, or reversal agents for drug overdoses, drug delivery, and small molecule-induced protein-protein interactions. It's a hard problem, illustrated by a beautiful paper here from the Baker lab with an impressive group of co-workers in which they endeavored to bind a very flat hydrophobic fluorophore shown here. And to be sure, they also designed for the first time a beta barrel of its type. But once they achieved that the barrel, it was massive virtual screening, dozens of proteins being prepared, ultimately finding some binders, and then extensive optimization by yeast display before they could break the 1 micromolar barrier. Could we go directly with a much more complex and purposeful molecule to bind with submicromolar affinity? That's the question. So how does one generally go about binding proteins or ligands in proteins? Well, you first have to choose a scaffold. And so many are in the PDB, and you can make a library of them. And then you just dock a substrate in, and then you design a sequence that stabilizes that, like this. So this happens to be neuraminidase, and here's a sialic acid derivative. So what's so hard about that? Well, there's 24 polar interactions, as well as second shell hydrogen bonds not shown in water. So that complicates life. Moreover, think about the core principle. This protein has evolved in detail to bind this, and not necessarily what you want it to, some other molecule. So we have to solve also the cooperativity problem, as I think of it, in that you're putting many, many ligands together, side chain ligands, that have repulsive interactions as well as favorable interactions with the molecule of interest. We simply sum up pairwise interactions, but we know in situations like this that they're rehybridizing, and once you get to the sub-angstrom level, it's really hard to simply sum up the pairwise interactions. Finally, there's a sampling problem here. Tenths of angstroms will have a large effect on the computed energy. So how do we do this? Well, how does nature do this? So nature always evolves in a given structural space, tertiary structure, by making very simple sequence changes. It doesn't worry about rotamers. It worries about the local sequence motifs. So give you an example. Here's a beautiful paper from Dan Toffick's lab in which he asks, how do proteins bind phosphate? And he sees a number of solutions. So if you have this tertiary structure motif here, or super secondary perhaps, and the right sequence, say a threonine right there, then that positions a phosphate precisely in space. Look at this. Here's a little bit of a helix. Here's the end of the helix and an arginine. That codes for put phosphate here. And in each case, you're seeing the placement of that function group relative to the protein backbone. And we call these positions that occur in conjunction with structure and a given amino acid sequence or two as privileged positions for binding chemical groups. And you get the binding of a full molecule when you sampled enough of these backbone sequence combinations to engage your molecule of interest, multiple functional groups. So an evolutionary inspired approach would be to choose a molecule of interest such as this. And then we're going to worry about bearing the most difficult groups. These are the ones that are hardest to take out of water and put inside a protein. We're going to have to get these just right. So we start with the hardest. And then we find privileged positions on a de novo backbone that can engage them. Now, to do this, we needed to invent a new element of structure, which we call the Vandermeer. Vandermeers are really related to rotamers in that if rotamers take a backbone and relate a side chain functional group to the backbone. So if I know the backbone and I specify a rotamer, I know where this carboxylate is. Vandermeer, on the other hand, starts with the backbone here. We don't worry about where the side chain is. Instead, we worry about where an interacting group is. So the way we find these is we identify all residues of a certain type, like aspartate here, that interact with a given functional group, in this case, a carboxamide in the PDB. Then we perform an all-by-all pairwise superposition of just the backbone there and the interacting group. And we cluster them with a reasonably tight RMSD of 0.5 angstroms. And for this work, what we've done is to just use the interactions that we see between side chains and other side chains as proxies for the interactions with ligands. That really improves our counting statistics. We can certainly extend this approach, for example, for phosphate, as we saw from Toffick's lab. Now, the reason this is so powerful for sampling is often you can have multiple rotamers that all conform to a single Vandermeer. So they all converge here and place a carbonyl at the same position, even though they have different rotamers. So these are different histidine carbonyl Vandermeers that are shown here. And these are what we use in the first steps of de novo design. Now, just as rotamers are backbone dependent, so too are Vandermeers. So here are a number of Vandermeers that were generated for carboxamide interacting with aspartic acid. Some are highly enriched in alpha helix, others in beta strands. And we were surprised to find that on the order of 90 clusters capture about half of the asp carboxamide interactions. This is a really reasonably small number, on the order of the number of rotamers that we would use to pack a core. Now, how does this differ from earlier work? Well, earlier work always used user-defined libraries of interacting groups. And there are many, many degrees of freedom that must be considered in creating such libraries. Now, look at what would happen, say, with an arginine. Here's the backbone. We can think of some of its rotamers shown here, far from complete. And then we can append to them functional groups. And what we end up with is sort of a mushroom cloud of functional groups, is seemingly suggesting that you can put a carboxamide anywhere. But if you look at the top Vandermeers, they're tightly clustered here, 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 here. So those are your privileged positions. Those are the ones that you really want to be sampling if you want to design a protein. Now, why is it that they're privileged? Well, often they're features that you really don't think much about. Here we're looking at how alanine can interact with a carboxamide in a helix. And what we see is this close approach of the carbonyl to the C alpha H in what is often called a C alpha H hydrogen bond. And that can occur between a turn of a helix or at consecutive positions. Another reason that we see such privileged positions is often we'll see the reinforcement of Vandermeers. Here's a carboxamide interacting with an asparagine. Let's put that on a helix. You can also, from a different position, interact with histidine and end up with a carboxamide in the same place, or a serine, or a carbonyl, or all of them at once. And so the reinforcement can occur not necessarily all of these all at once, but these are recurring patterns that tend to create more privileged positions. Of course, with a situation like this, now we have a unique position to attach a functional group, although we'll do it in a more cooperative manner. So as a case study, we started with this drug called Eliquis, or apixaban. And a cardiologist that I work with, Ethan Weiss, suggested it to us as a worthy molecule to try to bind. And this is a blood thinner. It inhibits factor 10A, and bleeding is a major problem from either overdoses or simply when one wants to reverse this to get patients into surgery. There is now an antidote, which is an inactivated 10A, but it was, and it is on the market, but it was experiencing problems with manufacturing for many years. So the question is, can we de novo design a protein that looks nothing like catalytically inactive 10A, but binds this with high affinity? And it is a stringent case. We have a total of 9 polar heteroatoms. We have 5 rotatable bonds, which cause unfavorable decrease in entropy upon binding. So we're at the edge of Lipinski's rules. And so how do we go about doing this? Well, we cut it into functional groups that we can relate to our Vandermeers. We create a small library of parametrically generated backbones. And then for each of those backbones, we iteratively paste on our Vandermeers and find where do they superimpose so that we can, with the functional groups of apixaban in a low energy conformation. We find a number of solutions, but this is one shown here where we've engaged all of our targeted groups. And now we have to ask, can we create the rest of the structure given those restraints? So we'll hold them in position and then use the core principle and variable backbone design, flexible backbone design to complete the design. we made 6 proteins, and 2 of them worked from the perspective of binding, one with a 0.5 micromolar KD and the other with a 5 micromolar KD. These were the only 6 that we made, and there was no need for optimization on phage or yeast or directed evolution to get to this point. Moreover, this was the first time where we were able to go directly from a design from a computer to a crystal structure, as you'll see, of the complex. The 4 that didn't work appeared to be molten globules, and when Nick ran folding simulations in Rosetta on them, they did not fold to the desired structure, so that could have been a tip-off for us. Now, the 2 that were designed have entirely different topologies and sequences. The only thing that they share are the keystone interactions that hold it into the bundle. The structure was solved at 1.3 angstroms resolution and agrees entirely with the design, with a few small exceptions. Now, it also is very specific. If we look at a closely related factor 10A inhibitor, it doesn't bind to it appreciably. And it's very sensitive to small changes to a pixaband structure. Nick also solved the structure of the apo form of the protein. And you can see that the cavity, here's the holo, here's the apo, and the cavity is pre-organized and is composed in the center is a elaborate network that holds all the polar groups together. And this network of water is released when the drug binds. Here, Nick has compared that water network from apo to the electron density from a pixaband in holo, and you can see how they all map to positions where we had polar interactions with a pixaband. I want to point out 2 interesting tyrosines, because these weren't included in the original design, and they came when we repacked with flexible backbone with Rosetta. And this one didn't agree very well with the design, and instead a water came in here, whereas this one worked out fine. Now, when Nick went back and looked for the Vandermeers to those 2 tyrosines in the designed backbones after flexible backbone design, he found that this one was considered a very good scoring Vandermeer. We used log odds to score the Vandermeers, and this scored well. On the other hand, the one that didn't work out so well that Rosetta had put in when we did mutagenesis, it didn't contribute much. This Vandermeer was not found in the entire database. So clearly we can see Vandermeers being useful not only for the initial design, but for vetting designs. So in summary, this is how we went about designing this apixaban binding protein. We followed the core principle to make sure that the core here communicates its information to the binding site here. Keystone interactions are very important, and they're hydrated in the apo state, and they release the water for a hydrophobic effect when the drug binds. So in summary, when we're thinking about small molecule binding protein design, we really let the desired ligand, the function, dictate the entire fold in detail, and then its sequence. We accomplish the hard part in the first step of computational design, which is cooperative binding of the polar functionality. And we let structural informatics drive sampling, but then physics-based force fields during refinement. And during COVID time, we have really begun devising new algorithms for recursive Vandemers. Let me finally finish by saying, why did we design this with a 4-helix bundle? It's simply because we really wanted to do something de novo that was completely parametric, and also 4-helix bundles aren't known to bind anything except for porphyrins and cofactors such as metal ions. So the ability to design something completely, or take a scaffold that isn't really intended for ligand design and show that it binds a drug, we thought would be a stringent test. Now, of course, these methods can be used for many other different structures. And let me just give you one virtual example in which we're looking at docking of glutamine to a periplasmic binding protein. Okay, and so what Nick's done is to cut this back to a polyglycine backbone, and he's going to ask, can I find in the invaginated regions positions that will bind the functional groups from glutamine? And so in particular, he's going to look for carboxamide Vandermeer. So where might he find it? Well, we go to the alpha hole and find where it can position carboxamides. We find a lot of them that superimpose. And along with that, we find which side chains superimpose them there. So this is identifying sequence as well as position of the carboxamide. That's a density into which we could now dock the carboxamide. We can now do this with the amine, and we'll use the lysine side chain as an ammonium surrogate and a carboxylate using the carboxylates of aspen glue as surrogates. So here's where we could bind a carboxylate. Here's where we could bind an amine. Remember, there's no sequence that goes into this. You have to find both the position to bind the amine as well as the sequence responsible for it. And now we dock in, and there's the sequence. We score the sequence just by log odds, the number of times we see these interactions being observed. And so the Combs finds the position for the ligand to less than a half angstrom accuracy and has 80% recall of the native interactions that position glutamine into the binding site. So with that, I think we've come sort of full circle with the Anfinsen hypothesis. This clearly shows how a tertiary structure not only specifies the sequence, but when you also think about its function, which is to bind this ligand, the sequence of the binding site itself. So I want to thank you all for your attention, especially also Nick for all of the work that I've described in the latter part of this talk, Yibing Wu throughout for help with NMR, and Joe for synthesis of some of the molecules. Okay, and so now I'm going to escape, and I'm not sure what happens next.
Okay, thank you, Bill. That was an excellent presentation. Beautiful work. So we can open the floor to questions. So if there are questions, please use the Ask a Question box. I think one question came in. Yes, there's already a question for you, Bill. So if you wouldn't mind reading it and answering that, that would be great.
I'm not seeing the question.
Okay, so click on Ask a Question.
Oh, what are the major issues of false positive, not enzymes detected by as enzymes?
That was a question for Joanna. So your first question is down the list. It starts with exciting results. Could you apply the Vandermeer protocol?
Exciting results. This is from Guto Rees. Whoops, it keeps jumping. Could you apply this VDM protocol to target abiological moieties, for example, pyridines and other NOS heterocycles? Yes, you've been reading our mind. And if you look at the paper we just published, we use biotin as an illustrative example. You'll notice that there are some unusual heterocycles in the pixaban. What's exciting is that proteins only have the 20 amino acids to interact not only with the ligands, but with themselves. And fundamentally, those interactions are pretty much the same. When we get into halo interactions, I think we have to also dial in information from the force field. But for most of the hydrogen bonding types interactions, electrostatics, I think we're pretty well calibrated. Thank you for that good question. How important is designing the molecule will be to the binding site, especially in bigger proteins? Oh, how important is designing how the molecule will get to the binding site? Yes. Okay, I think, you're going to have an access channel to it or breathing motions that allow it to come in and out. And I think when you're thinking about binding proteins, it's okay for it to be relatively slow and enfolded entirely by the protein. If we're thinking about catalytic proteins, enzymes, then we're really going to be really concerned about that access. How do they get in? How do they get out? And the dynamics of in and out. Let's see, there's one that may be for Joanna. How large should the training data set to make sure our model is correct? I think she answered that.
Yes. There's one that starts with, "This is fantastic."
Okay. This is fantastic. What do you think the right strategy is for defining Vandermeers for halogens? Yes. We have, for halogens, I think we just need to go to drug complexes. And there's quite a bit of information on halogen interactions, and we'll just have to construct those. Francois Diederich has, by the way, done a lot of that analysis already, and so we can incorporate that into our designs. Okay. In addition to affinity, association, dissociation kinetics often matter a lot. Do you think that binding kinetics will be designable in the future? I think I already addressed that issue. We are, in fact, dealing with that issue in our design of catalysts because we have to get things in and out quickly. On the other hand, if we're trying to make an antidote, it's great to just get it in, hold on to it, and then let it be filtered out in the kidney. So there's no one right answer. We want to have control. Do you envision using Vandermeers and scoring functions for docking and virtual screening? I think that would be great. As you saw, we were able to achieve high accuracy in terms of the docking pose. The way we use this scoring-wise right now is just a very primitive log odds score. So you would want to combine that, I'm sure, with the force fields that are already being used. I was wondering if you used in vivo or in vitro methods for translating the protein of interest. So all the proteins in the second half of the talk were bacterially expressed. We use a combination of chemistry, and expression for the other proteins that we talked about in the metalloprotein portion. The ability to synthesize proteins now that are on the order of 150 amino acids almost trivially through native ligation strategies also allows us to make all D versions of proteins. And this is really cool because you can escape immunogenicity this way. So if you wanted to make a protein that's a carrier, say, of a drug and target it somewhere, you don't have to worry so much about the immunogenicity. Do you think there might be a physics-based approach to predict Vandermeers for chemical groups that are underrepresented in the experimental data? Yes, we're very much working on hybrid approaches to do that.
And Bill, if I may, I'd like to ask a question as well.
Sure.
So the system that you created to allosterically regulate catalysis was very interesting. And I noticed that it decreased the activity. So can you envision, how modular is this? Can you take another one of your 4 helix bundles that binds some other molecule and then just graft it using the same strategy and hopefully get an allosteric activator from that?
I would think so. Our purpose in designing this, to be truthful, was really to put these 2 cofactors into close proximity. And that's another story altogether. But during COVID times, I got kind of curious. Hey, could you do a few experiments just to see if it's allosterically activated? Right. And so I think it should be very easy to either increase the dynamics because you binding somewhere makes it frustrated, right? Mm-hmm. In the interface region, 10, 15 angstroms away in this case, or to make it more favorable in the interface region. So.
Right. That'd be great. And another question is, when will we be able to get our hands on combs?
So there is a GitHub, and Nick Polizzi is updating it, working very hard on that, trying to make it very, very user-friendly and have the libraries available to you. I think he's shooting for November, but there is something, I think, available already on GitHub.
Okay, great, thanks.
And I think, email Nick.
Right. I think there's going to be, after your talk, a lot of people will be interested in using it. Okay, there was a question that I think you missed. The question is, exciting results on the coupling of structure packing with binding. Can you specify the nature of the physics-based force fields for guiding refinement?
Currently, what I would like to be using is purely physics-based in the future. What we're using at this point is Rosetta.
Okay. Okay, great. Are there any final questions? Okay, great. If not, then I'd like to thank both of our speakers for giving excellent presentations. There was a lot of nice discussion. And I remind you that we have 2 more webinars to go, one in late October, the other one in late November. So please check the PEDS website for the information. And also, I would like to remind you that if you want to rewatch this, it is on YouTube, so you'll be able to go there. And I will post the link for the YouTube presentation on the PEDS Twitter feed. So if you're not following us, you can follow us there. All right, thank you, everybody. Have a nice day.